Hello, my name is Aiden Udan Dunstan, and my matrix number is 97977, and I am from Subgroup 7, Group 2.
Hi, my name is Asma Binti Draman, and my matrix number is 98267.
Hello, my name is Deirdre Ayen, anak Judas, with the matrix number of 98698.
Hello, my name is Edna Alicia, anak Chimiam, with the matrix number of 98765. The objective of this practical are to perform simple dilution of glucose samples for sugar analysis using the DNS method, to determine the absorbance spectrum of glucose solutions at different concentrations with a UV visible spectrophotometer, and to plot a glucose standard graph with its corresponding linear equation for accurate estimation of glucose concentration in the unknown samples.
The following are the materials for these experiments. Firstly, 6 clean test tubes were prepared and labeled from number 1 to 6, where test tube 1 were reserved as a blank. Then, the following volume of glucose solution and distilled water were pipetted into respective test tubes. This was done in duplicate. After that, DNS reagent of 1 ml were added to all of the test tubes. After all of the test tubes were filled with all of the solutions, they are shaken well using a vortex mixer. Then, the test tube were placed in the boiling water bath for 5 minutes. After 5 minutes, the test tube were taken out of the water bath to cool until reaching room temperature. When cooled, 1 ml of Rochelle salt were added to all of the test tubes. Then, around 2 ml of each of the solution were transferred into the cuvette. The optical density of the colored solution was read at 575 nanometer using test tube 1 as a control. The reading were recorded and the relationship between the optical density and the concentration of glucose solution was plotted.
In conclusion, for practical 2A, we prepared a series of glucose standards ranging from 0 to 1.0 grams per liter and measured their absorbance at 575 nanometers after reacting with DNS reagent. The absorbance values increased proportionally with glucose concentration, creating a clear linear relationship that confirms the Beer-Langmuir law. Our standard curve equation y equals to 1.37x plus 0.1167 with an R-squared value of 0.9358 demonstrated a strong correlation between concentration and absorbance. This standard curve is essential because it allows us to determine unknown glucose concentrations for practical 2B. Overall, this practical showed that the DNS method combined with spectrophotometry is a reliable approach for quantifying reducing sugar.
For this experiment, one of the main things we learned was how to properly use a micropipette. Since we were working with very small volumes to prepare different glucose concentrations, it taught us the importance of accuracy and technique during sample preparation. We also gained a better understanding of how the DNS method works. It helped us see how reducing sugars like glucose react with the DNS reagent to produce a colored compound and how that colored intensity can be measured to reflect sugar concentration. Another key skill we picked up was using Microsoft Excel to process our data. We used it to calculate average absorbance, find standard deviations, and generate a glucose standard curve. This made it easier to interpret our results and apply the linear equation later on. Lastly, We learn how crucial timing and temperature control are during the boiling and cooling steps. Any variation could affect the color development, so we had to make sure everything was done consistently across all test tubes.
As for the challenges, first one is uneven sample amounts. It was challenging to ensure that each sample had exactly the same quantity, which could have affected the consistency of the observation. As for the limited time for reaction and observation, time constraint during the lab session made it difficult to observe any changes that might have occurred more clearly if more time was allowed. The first one is repeat test with triplicate. Run each test in triplicate to ensure the consistency and reliability of the results. This would help to reduce random errors and strengthen the accuracy of the observation. Improve sample preparation. Use more precise measuring tools such as micropipettes or digital balance to prepare equal and accurate amounts of each sample. This will make preparation between samples to more be valid and fair.
For other suggestions, we can use more intermediate concentration points for the standard curve, including additional concentrations like 0.3 gram per liter, 0.5 gram per liter, or 0.7 gram per liter between the existing values would help refine the calibration curve. This gives a clearer picture of the linear range and can reduce error when estimating unknown concentrations. Next is monitor and standardize boiling time more precisely, using a timer to ensure that each test tube is boiled for exactly the same duration could improve consistency in color development. Any variation in heating time might affect the intensity of the color product and lead to inconsistent absorbance readings.